Thank you, Simon, for the introduction. So, first of all, I would like to give you a brief overview about today's agenda of that bioscience webinar we are giving. And we will start with taking a deep look into one of nature's most important classes of biomolecules, that is proteins, and we'll learn how IR is utilized to study them. Then we will follow up with a few IR fundamentals. How is IR working in general and how is it applied on proteins to study them? Just to follow up with other biomolecules that can be studied by IR and also much more complex materials such as biofluids and multi-component liquids. After seeing a lot of very interesting applications, Michael will introduce you with a IR measurement equipment that can be utilized to do all that research. And then we will take a look at, let's say, complex biomaterials and how they can be analyzed in a microscopic manner, how IR can be used to do chemical imaging on biomaterials and biological tissues. And again, Michael will give you some introduction in with what equipment you can do such research. Finally, you may have a couple of questions and we will try to answer them in most helpful manner. And now I will continue with our first topic, the analysis of proteins. So proteins, what are they? They are basically macromolecules that are formed by a sequence of amino acids that are bound to each other by a peptidic bonding. And proteins provide very important functionality in bioorganisms and very multifold. So they can act as enzymes, that means they catalyze chemical reactions within the body. They protect the organisms, that is, for example, by antibody proteins, so they are part of our immunogenic system. They work as messengers to do signal transmission. They also are part of the body shape, so they are structural components that form a living being. And finally, they also work as transporters to deliver molecules. Just think of hemoglobin bringing the oxygen to the right place in the body. This wide range of functionality is always related to the actually present three-dimensional structure of such a protein. And if that structure is not present, the function will not work as expected. And here at that point we may take a look at how this structure looks like, because it's a quite complex thing. So first of all, we have the primary structure, which is the specific sequence of amino acids within the polypeptide chain of a protein. This chain forms a helix or strands or stays unordered, and that is what we refer to as the secondary structure, which again forms a three-dimensional form in space, and that is called the tertiary structure. Finally, proteins can be present as monomers, but also form multimeric units, oligomers, and that is what is called the quaternary structure. So you see, the protein is a quite complex molecule, and there's no single analytical method that can give you a deep insight into all these structural elements. So it is a wide range of methods which is applied to get a full picture of protein structure, protein functionality. And within this method portfolio, infrared is particularly sensitive to look at the secondary structure, but as the secondary structure also is like affected by tertiary and quaternary structure and changes of those. Yeah, in some way the infrared spectroscopy is a tool that allows you in the so-called higher order structure of a protein and detect changes within this. As we have seen that proteins have so many different functions, it is not too hard to imagine that they are relevant for many academic disciplines. That is, of course, biology, but also in pharmacy, food science and material sciences. Yeah, proteins have a high relevance and are studied by researchers. And in the following, I will present you some of the applications that have been performed in the past years, and always refer to recent publications that are related to the topics I'm presenting. And don't be worried that you need to write down the title of the publications if you're having a deeper interest. We will provide, together with the recording, a listing of all the references that are shown during the webinar. In biology, it is about the determination of the relation of protein structure with the biological properties of a protein. So this can be like the immunological activity of a certain protein when it acts as an allergen on organisms. In many cases, we want to understand protein reactions. So how does a structure of an enzyme change when it is activated to do a repair of a damaged DNA? What are the modifications of the protein during the process of photosensing or photosynthesis? These processes happen on a very different time scale. The DNA repair, it takes minutes to hours until this entire activation process is completed, while photosensing takes place in the sub-second region. So we see it here indicated on that graph on the left-hand side. And IR spectroscopy delivers methods that allow you to follow such processes, no matter if they are like in a larger time scale or extremely quick. The capability of proteins to protect and even heal us from diseases makes them extremely interesting for pharmaceutical research. So in the past decade, many biopharmaceuticals have been released and are used as therapeutics. Among those, there's many monoclonal antibodies, but also insulin for the treatment of diabetes. The contribution of IR to study those therapeutic proteins is to characterize their stability against external stress factors such as heat or shear forces or light irradiation. Also, the protein content within vaccines is examined by IR spectroscopy, and here it is the interaction with adjuvants that is of particular interest. So how does the protein structure look like when the protein is absorbed to a certain adjuvant such as alum, and here IR can deliver the required information compared to the purely solved protein. Proteins are also a vital part of our daily nutrition, and they are present in higher or lower amounts within the food products we eat each day. For that reason, it is interesting for food sciences and technology to determine the relation of the structure of those food proteins with the resulting properties of the product. Such properties may relate to health aspects, such as the allergenic potential of food proteins, but also to functional properties that are wanted for a food product, such as the mouthfeel of a yogurt or some other milk product. Therefore, food technologies study the impact of the food processing parameters on the protein structure and how that relates to the final food quality. So in the example shown below, we see a milk protein that has to be processed at a certain temperature to result in exactly the right solubility and emulsion that is wanted for the final product. It is a similar approach that is followed by material sciences. Here, the structure of a natural biopolymer like silk is studied, and based on the learnings, it is tried to modify its characteristics in a wanted manner. In case of the silk, there's a protein inside, the fibrin, which forms better sheet-rich fibrils that are giving the strength to the thread of the silk. Here in that study, it could be found that by adding another biomolecule, chitosan, a new composite was formed with even a higher beta sheet content, which increased further the strength of the resulting material, and that bears new potential for developing biotissues that can be used for medical applications and also for new textiles with different properties than were available so far. Now you have seen the first set of applications of IR spectroscopy for proteins, and you may wonder, how does that method work in general, and what does it tell us actually about the proteins, and is it all IR can do, or is there more? So let me show you in very brief manner the principle of IR spectroscopy. So the principle is that the IR light interacts with chemical molecules and induces vibrations of the chemical bonds inside these molecules, which leads to an absorption of energy, and this absorbed energy is measured with our IR instrument. As a result from that absorbed and measured energy, we get an IR spectrum of the individual sample that we've measured. So here there's an example shown for the water molecule. Now you've seen already a water spectrum, but what do vibrations tell us about proteins? So first of all, the IR spectrum of a protein allows us to get information about its secondary structure. So we can learn about the structure of unknown proteins and use the spectrum to check the conformational integrity of that protein. We also will recognize if there's certain conformational changes, and this helps us to study the structure-function relation as we've seen in the examples before, and also to control if a protein is stable or if there's any denaturation process ongoing. Finally, it allows us to study protein dynamics. So if we induce protein denaturation by stress factors, this can be followed online, and we can also see protein folding processes like they occur in reactions, also as shown earlier in this presentation. Now we've seen what kind of analytical information can be obtained from the IR protein spectrum, but we also want to take a look at its general characteristics and how it compares against other analytical approaches. The IR method allows a quick insight into real-life samples, Protein formulations can be analyzed as they are, and results are available in the order of minutes. The method delivers full conformational information. It is sensitive not only to helices but also to beta sheet structure, and it allows the differentiation between inter and intramolecular beta sheets. IR is universally applicable, so it can be applied on proteins and peptides, on solutions, opaque samples, suspensions and solids. There's no limitation of specific buffer components or excipients. All these properties make it a unique method that can be used with high throughput and virtually on any kind of protein material. So now we've seen a lot about protein analysis, but IR spectroscopy actually delivers much more information because, all kind of chemical molecules have excitable bonds, which means that the IR measurement delivers a unique spectral fingerprint of each chemical component. And from this chemical fingerprint, we can obtain information about a substance or sample's identity, about quantity of components, about conformation, stability, kinetics, composition, and parameters which are correlated to the above. Yes, and the method is actually applicable not only to isolated but also to complex biochemical samples, and this is what I would like to take a look on in the next section of this webinar. Now, to what other kind of samples than proteins do we want to take a look now? That is natural products, biopharmaceuticals, and last but certainly not least, body fluids. Natural products, that is nature's chemistry kit. It is interesting to identify and characterize chemical components that can be isolated from natural resources such as microorganisms, plants and fungi, and even animals. And we want to learn what kind of natural chemicals are in there and reveal their structure and characterize their properties. So, nature is full of chemical components which are synthesized by nature itself. So, scientists are now extracting these individual substances from microorganisms, from plants or fungi and animals, and then they want to identify and characterize them, where IR spectroscopy is a very suitable tool. By using vibrational circular dichroism, it is even possible to determine the absolute structure of molecules that are chiral, so it is possible to determine the exact enantiomer that comes from the natural resource. So far, the applications that have been shown in this webinar always were focused on the analysis of a single substance or component of the sample. But IR can also be utilized for multi-component analysis in samples such as biologics. Here we see, of course, the active pharmaceutical ingredient, the protein, but next to it also the excipients, detergents, buffers and possible impurities that are present in a certain formulation contribute to the IR spectrum. And from that single spectrum we can get qualitative and quantitative information about individual components and the entire composition. So in general, the IR spectrum allows us to take a holistic view on complex sample materials. However, Biopharmaceuticals are samples that still have a very defined composition. But the same spectroscopic approach also can be applied on even more complex and variable materials such as body fluids. So that might be blood or urine or saliva, and the infrared spectrum of such a body fluid shows its entire biochemical composition. So there's information of all the individual proteins within the sample, of the lipid, the phosphates which come from the DNA, carbohydrates, and so on, so of all biomolecules that are present in such blood or urine sample. And based on that complex spectroscopic fingerprint, we may get indications about the health condition of the individual from which this sample has been taken. In this regard, I would like to present the results of a study where blood samples were taken from many individuals and measured by IR spectroscopy. One of the findings was that the characteristics of the IR spectrum is quite person-specific. And the next finding also was that these characteristics stay very stable over longer periods of time, so even over the course of weeks and months. Only for some individuals there was at a certain time point some change of that IR fingerprint observed, and that provides a perspective that the IR spectrum of blood might be suitable for continuous health monitoring and also allows us to detect specific medical phenotypes of persons. There also have been many studies where body fluids were measured by IR spectroscopy with the goal to detect diseases at a very early stage. The type of samples that were used were mainly blood-based, but also bile, urine and saliva were utilized, and in these studies the proof of principle could be made that IR is capable to detect certain cancers, diabetes, arthritis and other diseases based on the spectral fingerprint. And for that reason, today there are the first commercial IR diagnostic products under development. Now, as you have seen all these interesting applications, I'm certain you also want to learn what kind of equipment is required to perform the measurements. And for an introduction of the IR instrumentation, I will hand over to my colleague Michael.
Thanks a lot, Martin, for the handover. Well, all the data introduced to you from Martin was measured with an IR spectrometer at heart. This heart is our FT-IR analysis hub in VINO, which I'm going to present in the following slide. Building upon VINO spectrometer, we offer various solutions for different measurement tasks. Especially important for this webinar is the ConfortCheck system for measuring proteins in liquids, as well as the HDS-XT extension for increasing the throughput of your measurements of solid samples. Let me also show you more options to really satisfy all needs which may come across your working day. You have the best idea of VINO when you think of this spectrometer as a versatile, powerful and flexible analysis hub. VINO is the device where the actual interferometer for measuring the spectra of the samples is located. It absolutely is the premium device what just makes you proud owning it. Compare it to any other competing spectrometer in the market and you will immediately see and feel the superiority of VINO in terms of build quality, operability and design. It comes with the best components for highest performance and robustness. Just take our interferometers rock solid or integral as examples. They are self-aligning and at the same time wear-free, guaranteeing highest robustness and fail-safe operation at all times. VINO gives you lots of output for your input. It has the capability to add a parallel measurement channel in the device itself as well as input and output ports to extend your IR analysis system with more measurement methods. It does not stop at the hardware. We have lots of software, algorithms and libraries available for providing comprehensive results in a fully automated mode. VINO also provides you with the best usability and user-friendliness. All operation and analysis is automated and controllable in our software Opus. You can even get a touch panel PC with the dedicated touch software for more convenient operation without the need of any peripherals as shown in this picture. Also, the core specifications, such as spectra acquisition rate for transient processes, the spectral range or even more specs can be adjusted to your requirements. Furthermore, you can use various accessories and external modules to extend the system for a multitude of measurement modes. Those extensions, together with the configurable core specs, can be combined and used in a flexible manner, making VINO really your powerful IR analysis hub. Finally, VINO always offers the option to upgrade to better system specifications or to add more features and extensions after the system was purchased at any time and at any place. This guarantees that VINO is the right IR analysis workstation that can also deal with your future challenges. Based on VINO, we offer the turnkey solution ConfortCheck for protein and biofluid analysis. As the name suggests, it is especially suited for analyzing the conformation of proteins. Our solution ConfortCheck mainly consists of those 5 items. The first item is our spectrometer VINO, which I just introduced to you. Along with the spectrometer comes the accessory AquaSpec, which can be mounted in the main compartment of VINO. AquaSpec will measure your sample solved in water. It is the right accessory if you want to determine the secondary structure of your proteins, analyze the conformational stability, or measure a sample concentration in a fluid. ConfortCheck also comes with all software methods needed to quantify and evaluate the concentration and the conformation of your protein sample. It is able to cover the full spectral range from 4000 to 950 wave numbers. This is very important as it is the only way to enable meaningful measurements and give control of signal validity. The next accessory is the BioATR2 cell. BioATR2 is made for the investigation of thermally induced structural changes as well as aggregation and precipitation analysis. As for AquaSpec, BioATR2 cell is measuring protein samples in water. Compared to AquaSpec, BioATR2 is measuring the sample via attenuated total reflection and not transmission. The cell is detachable for easy and convenient cleaning. The full biorelevant temperature range from 4 to 95 degrees Celsius is covered. The needed temperature ramping can be done fully automated via our software Opus. Also here, the measurement covers the range from 4000 to 950 wave numbers. ConfortCheck also comes with an additional ATR channel for solid samples, which we call the Transit ATR. It is a perfect extension to VINO, where the main sample compartment already contains an accessory for fluid analysis. With the Transit ATR, you can do parallel measurements of solid samples in the very same spectrometer without touching or influencing the setup in the main compartment. This enables 2 measurement methods being ready in parallel at any time. The switching between the methods can easily be done with a click in our Opus software. Transit ATR is very robust due to its internal diamond crystal and covers the full MIR spectral range for comprehensive measurements. This makes it perfectly suited for any routine application, such as identification or incoming inspection. You can also use it for testing of intermediate and final goods, or for fail analysis by chemical identification of separated precipitates. The next important item of our ConfortCheck solution is the System Validation Package. The System Validation Package includes everything to make the system with all its accessory compliant to the US 21 CFR Part 11 regulations, various international pharmacopoeia, and current goods manufacturing practice. Along with that comes a system validation manual to assist and conduct the validation procedure, where all needed test reports are generated automatically. These 5 items make ConfortCheck a true one-stop shop for protein analytics. There is even more to learn. Please let us know in case you need more information. An important extension to our analysis hub in VINO in this bioscience context is our HTS-XT. HTS-XT is our extension to increase the throughput of your analysis. It features automated measurements in either transmission or diffuse reflection mode. It is capable of handling up to 1,536 samples in one go. For this, the samples need to be in solid form and are filled in standard, reusable microplates to be inserted into the system. There is still much more to learn about the capabilities of VINO in the bioscience context. Besides ConfortCheck and HTS-XT, let me just briefly touch the most important ones. VINO is capable of doing time-resolved measurements, which are needed, for example, to analyze and understand chemical reactions of proteins. You can reach a time resolution of 15 milliseconds for free-running experiments with our rapid scan technique. For repeatable measurements, you can even get incredible time resolutions down to just 4 nanoseconds with our option step scan. There are also more extensions which might be of use for your highly sophisticated analysis. Just to name a few, we can supply the external modules PMA50 for VCD measurements, the RAM2 for FT-Raman measurements, or our microscope Hyperion II for spatially resolved measurements. The microscope Hyperion II actually is a very good chance to pass the word back to Martin, as we are now leaving the macroscopic world and turn towards applications where microscopic and spatially resolved IR analysis is used.
So far we have just presented applications that were performed on macroscopic samples, but IR analysis can also be performed with high spatial resolution. And here, in the field of biosciences, it is certainly the chemical imaging of complex biological materials and tissues which is the most exciting and promising application. The chemical imaging of biological materials is particularly relevant to understand biological structures, but it is also performed in material sciences, and the most exciting potential lies in medical research. Tissues are biological materials with complex structures that are made from biochemical components that are inhomogeneously distributed in that tissue material. By applying IR microspectroscopic imaging on such materials, it is possible for biologists and material scientists to study the spatial distribution of those biochemical components. From the chemical images, it is now possible to relate between the material composition and its properties. So here there are examples of chemical images shown that were taken from silk tissue, where the chemical images show the distribution of the protein content as well as on carbohydrates. And on the right hand side we see also the areas in the silk that show a particularly high beta sheet content, which indicates a high amount of fibrils and is a property of the silk. The same method, of course, can be not only applied on such silk webs but also on plant materials to study the cell composition. In medical research, the approach is a different one. Here the IR images of clinical tissue sections are used to develop a spectral histopathology method. The hyperspectral tissue image is analyzed by modern machine learning algorithms with a goal to differentiate between healthy and diseased tissue. The upper image on the right shows the visual microscopic view of a stained tissue section. In the middle, the complete IR image is displayed, and the one on the bottom selectively shows the area that was detected to be cancerous. Bruker is having all the right IR imaging technologies to generate such impressive results. So for now I will pass over to Michael to provide you a nice overview about our FT-IR and QCL imaging microscopes.
Thank you, Martin, for showing these impressive examples. Before we close this presentation and step into the question and answer session, I will now briefly introduce the measurement equipment needed for making these spatially resolved IR measurements. We offer two microscopes for that. The first I want to present is the Lumos II. Lumos II is a very user-friendly and easy-to-use microscope. This microscope does not require an VINO spectrometer to do spectroscopy compared to all other devices I presented here. An interferometer for getting the spectra is already integrated in this beautiful product. Because of that, and also by design, it has a very small footprint fitting on every lab table or bench. Lumos II is fully automated and controllable via our software. This includes, for example, the sampling stage or even the condenser. As you can see, there is no single button located on the device needed for operation or configuration. Lumos has transmission, reflection, and ATR measurement methods ready for you. Regarding detectors, you can choose between a single element detector or a focal plane array detector to capture larger parts in a single measurement of your sample in a shorter time. Furthermore, Lumos is ready to be operated in a validated environment and, just like ConfortCheck, compliant to all major regulations. Besides Lumos II, we also offer Hyperion II as an extension to VINO, which I'm going to present in the following slide. So anybody who needs cutting-edge technology for faster speeds, best performance and highest flexibility in microscopic IR analysis will be happy to hear that we just recently launched our new Hyperion II microscope. It is coupled to VINO and uses its broadband light source and interferometer for making microscopic FT-IR analysis. In addition to that, Hyperion II has the option to use IR laser imaging based on quantum cascade lasers for generating spatially resolved spectra. You don't have to choose for one of these options to generate a spectrum. By doing a single measurement, you will get a dataset including information from both FT-IR microscopy and infrared laser imaging if needed. IR laser imaging makes your chemical analysis about 100 times faster compared to conventional IR microscopy. You can use that speed for ultra-fast image generation or even for infrared live view. The resolution of the images you get are only limited by diffraction, and coherence effects coming from the laser illumination are suppressed. Just like Lumos II, the measurement methods ATR, transmission and reflection are available. As I said, infrared laser imaging is an option to Hyperion II. You can also use it without laser imaging with a single pixel detector or a focal plane array detector. However, I strongly suggest that you don't miss the IR laser imaging feature and its capabilities. So please check our website and media channels for more information and use cases. With all that being said, we move forward to the question and answer session with a few words from Simon.